Hello friends, how are you? Hope you are doing great in your studies. I'm Preeti Rana, and in this video we're going to study about biotechnology and its applications. Before starting the topic, let us discuss what is biotechnology exactly is. So, biotechnology is that branch of science that uses microorganisms, animals, plants, cells, or any part from the body or their components to generate the products and services which are useful to the human being. As we have discussed about biotechnology in my previous video, the link of that video has been given in the description box. But for that overview, as you can see, here is the donor DNA or the DNA from which we will take the gene of our interest, and this is the vector DNA which will act as a way to transfer gene of our interest from one cell to the another cell. The donor DNA has been cut down into various fragments using the restriction endonuclease enzymes, and likewise vector DNA has also been cut down into fragments. Then both the vector DNA and the fragment of donor DNA has been joined together using the ligase enzyme, and that hybrid DNA has been incorporated in the host cell where it multiplies itself to form its clones. Biotechnology deals with the industrial scale production of biopharmaceuticals and biologicals using genetically modified microorganisms, fungi, plants, or animal cells. The application of biotechnology includes therapeutics, diagnostics, production of genetically modified crops for agriculture, processing of food, bioremediation, treatment of waste, and energy production. So techniques have been developed to produce rare and medicinally valuable molecules, to change hereditary traits of plants and animals, to diagnose diseases, to produce useful chemicals, and to clean up and restore the environment. This flowchart shows the various sectors in which biotechnological applications have been implemented. In the stream of agriculture, for the production of genetically modified plants, hybrids, micropropagation, for the formation of biofertilizers and biopesticides, and the improvement of animals. In the case of healthcare, for the formation of recombinant DNA products, vaccines, diagnostics, monoclonal antibodies, and tissue culture. In case of environment, soil and water remediation, biosafety, and genetically modified organisms. In case of industry, for the production of enzymes, fermentation based products and food, biotech instruments and equipments, bioenergy, biofuel, bioinformatics, biomining, and many more. Biotechnology basically has three research areas. First is to provide best catalyst in the form of improved organisms using a microbe or pure enzyme. Second is to create optimum conditions through engineering for the catalyst to act. And third is downstreaming process technologies to purify the proteins and the organic compounds. So this diagram shows the process of biotechnology and its applications in various sectors. This is the bacterial cell having the plasmid, then the plasmid from the cell has been isolated, and this is another cell from which we will take the gene of our interest. Both the plasmid and the gene of our interest have been joined together using the ligase enzyme, and that recombinant DNA has been incorporated back in the bacterial cell where it multiplies to form its clones. Such copies of genes have been used for the production of pest-resistant plants and for the treatment of toxic waste in the environment, and the proteins of form have been used to dissolve blood clots in the heart attack therapy. First of all, we will discuss about the application of biotechnology in agriculture. It involves three options. First is agrochemical based agriculture, which involves the use of pesticides and fertilizers. Second is organic agriculture, which does not involve use of any kind of chemical. And the last one is genetically engineered crop based agriculture, which is using the genetically modified crops for increasing the crop production as well as the quality of the products formed. This is known as green revolution, which increases the crop production. This includes improved crop varieties, agrochemicals, that means chemicals, fertilizers or pesticides, and third is better management practices. We have used the words genetically modified organisms. What does it mean? Genetically modified organisms are the plants, animals, bacteria, fungi, or any organism whose genes have been altered by genetically engineered processes. And the genetically modified crops are the DNA of which have been modified using genetically engineering methods to increase their crop production and to develop the resistance against the pests so that we can get the crop yield without the use of any kind of chemicals. The modification in the genetic structure of the plants let the crops become more tolerant to abiotic stress like cold, drought, salt, pH, temperature, etc. and the dependence on chemical pesticides they reduced greatly. That is, pest resistant crops have been produced which will help in the reduction of the post harvest losses. The efficiency of mineral usage increased in plants, so which will in turn prevent the loss of soil fertility, but this will greatly help in increasing the nutrition value of the food. Application of biotechnology in agriculture includes the production of pest-resistant plants which will effectively reduce the usage of biopesticides, leading to the production of organic crops. For this, Bt toxin, which is produced by a bacteria, is used for the production of resistance in plants against the insects in effect created by pesticides. For example, Bt cotton, Bt corn, golden rice, tomato, potato, soybean, etc. Bt cotton is created by bacterium Bacillus thuringiensis, Bt in short form, hence known as Bt, and the toxins of form is known as Bt toxin. This bacterium produces a protein which kills certain insects like lepidopterans, coleopterans, or dipterans. Bacillus thuringiensis form protein crystals during the phase of their growth, and these crystals contain a toxic insecticidal protein. Bt toxin protein exists as inactive protoxin in plants. When an insect ingests the plant, the inactive form of toxin is converted into the active form of toxin due to the alkaline pH of the insect gut, which solubilize the crystals. The activated toxin binds to the surface of the midgut epithelial cells of the stomach and create pores that causes cell swelling and lysis. Lysis means breakdown, leading to the death of the insect. Most of the Bt toxins are insect group specific. The toxin is coded by the gene named CRY. So the protein encoded by the genes CRY1AC and CRY2AB controls cotton bollworms, and the proteins encoded by the gene CRY1AB controls corn borer plants. This diagram shows the production of genetically modified pest-resistant plants. The insecticidal gene have been created by the recombinant DNA technology. For this, the gene encoding for the toxin protein have been cleaved from the Bacillus thuringiensis, which will be then joined with the vector using the ligase enzyme, and that recombinant DNA will be incorporated in the plant cell, which is taken in the culture medium. The plant cells which have taken the insecticidal genes will be separated apart, and those plant cells containing the insecticidal genes will be propagated further to form the pest-resistant crop. And this here shows the insect and in result have died off due to the toxic. Next example for the production of pest-resistant plants is a resistant in tobacco plant against the nematodes. A nematode, Meloidogen incognita, infects the roots of tobacco plants, which greatly reduce the production of tobacco. Agrobacterium vectors are used to introduce nematode-specific genes in the host cell. It produces both sense and antisense RNA strands in the host cell. The two RNA strands being complementary to each other, they join together to form double-stranded RNA or dsRNA that initiates RNAi, that means RNA interference, which will silence means suppress the specific mRNA of the nematode. RNA interference process is used as a cellular defense. It involves suppressing a specific mRNA due to a complementary double strand. It not only occurs in nematode only, it occurs in all the eukaryotic organisms as a method of cellular defense. mRNA interference. It is a biological process in which RNA molecules inhibit gene expression or translation by neutralizing targeted mRNA molecule. Double-stranded RNA binds and prevents the translation of mRNA, and the source of that double-stranded RNA could be from infection from any virus containing the RNA genome or from any mobile genetic elements that replicate by RNA intermediates. As the parasite cannot survive in transgenic animals, so whenever they attack any plant, they die off, and thus the plant got protected from the parasite. The flow chart here shows the production of transgenic plants. The first step is the isolation of useful gene using restriction endonuclease enzyme. Second involves transferring of the desired gene to form the recombinant DNA molecule. Third is transfer of that recombinant DNA molecule to the target cell. Fourth one is detection of the cell for transformation. Fifth is selection of the cell which has taken up the desired gene or the pest resistant gene. And the last being the regeneration of plant from the transformed cell to form transgenic plants. Next, we're going to discuss about application of biotechnology in medicines. The biotechnology application is very important in healthcare because it allows for the mass production of safe and more effective medicines. It prevents undesirable immune responses which are common with medical products from non-human sources. Non-human sources means from animals or from any other organisms. Made immense impact in the area of healthcare by enabling the mass production of safe and more effective therapeutic drugs. Recombinant therapeutics do not induce unwanted immunological responses as in case of most of the products which are isolated from non-human sources. Currently, about 30 recombinant therapeutics have been approved for human use all over the world, and in India, 12 out of these have been marketed. First, we're going to discuss about genetically engineered insulin. Insulin is the hormone which is secreted by the pancreas, which regulate the amount of glucose inside the human body. The reduction in the amount of insulin inside the human body leads to the diabetes. If the human body is unable to produce enough amount of insulin to regulate the amount of glucose inside the body, then the insulin need to be taken from the external sources. In the earlier times, the insulin was extracted from the pancreas of slaughtered cattle and pigs, and this caused allergy and various other reactions. Talking about the structure of insulin, it consists of two polypeptide chains, chain A and chain B, which are linked together by disulfide bridges. In mammals, insulin is synthesized as a prohormone which needs to be processed before it becomes a fully mature and functional hormone. It contains an extra C-peptide chain between chain A and chain B. Diagram. This shows the production of mature insulin from the prohormone. Here you can see the gray color represents C-peptide, the blue color chain A, and the green color chain B. Both chain A and chain B, they are joined together by disulfide bridges. So for the production of mature insulin, the peptide C needs to be cut down from the chain A and chain B. So upon cutting down the C peptide, it becomes free from it, and the peptide A and B, they are joined here with the disulfide bridges, and this collaborative will form mature insulin, which will help in the regulation of amount of glucose inside the insulin. C-peptide is not present in mature insulin, and it needs to be removed during the process of maturation of insulin. So in the earlier time, the challenge was to produce insulin using recombinant DNA technology in the insulin in the mature form. American company Eli Lilly in 1983 prepared two DNA sequences which were complementary to chain A and chain B of human insulin, and introduced them in plasmids of E. coli to produce insulin chains. For this, the chains A and chain B both were produced separately. They were extracted, then combined together by the disulfide bonds to form mature human insulin. This diagram shows the production of human insulin in the lab conditions. For this, first we have to take pancreatic cells which are good to produce in human insulin. Then the plasmids which are extracted from the bacterial cell. The gene which is producing insulin is combined with the vector to form recombinant DNA. That recombinant DNA is incorporated in the bacterial cell, where it multiplies to form its clones. Here you can see the bacterial cells having the recombinant DNA. The yellow dots over here, they represent human insulin. Recombinant bacteria multiplying and producing human insulin in the fermentation tank. Then the insulin is extracted from the fermentation tank and purified by downstreaming processes, and then is collected in a separate bottle which will be then marketed. Second application of biotechnology in medicine includes the production of vaccines. These vaccines are produced by genetic engineering, and such vaccines are known as recombinant vaccines, subunit vaccines, or second generation vaccines. For example, hepatitis B. These recombinant vaccines are of two types: protein vaccines and DNA vaccines. Protein vaccines use specific proteins which are produced by recombinant DNA in vaccine, and the DNA vaccines use genetically engineered DNA to be injected as vaccine to produce an immunological response. That means that DNA is directly injected inside the body, where that DNA expresses itself and forming the immunological response. Hepatitis vaccine contains viral envelope protein, hepatitis B surface antigen. This gene is isolated from the yeast vector. Encoding genes isolated from the pathogens are incorporated in the plants, where they express to produce antigens. So such vaccines which are incorporated in the plants and which we can consume by eating are known as edible vaccines. The next application of biotechnology in medicine is gene therapy, which is the collection of methods that allows correction of gene defects which are diagnosed in child or embryo. Genes are inserted into the person's cell and tissues to treat a disease. Correction of genetic defect involves delivery of a normal gene to the individual in place of the defective gene, so that the normal gene can take up the function of the non-functional gene. The first gene therapy was given to a 4-year-old girl with adenosine deaminase deficiency (ADA) by M. Lees and W.F. Andresco in 1990. ADA deficiency is caused by the deletion of gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation and enzyme replacement therapy, but they are not completely curable. In the first step of gene therapy, lymphocytes from the blood of the patients are grown outside the body in the culture medium. Complementary DNA for ADA deficiency is introduced in the lymphocytes. As the lymphocytes, they are motor, so patients need periodic infusion of some genetically engineered lymphocytes side by side. If the genes isolated from the bone marrow cells produce ADA is introduced into the cells at the early embryonic stage, it could be permanently cured. The other diseases that can be treated by gene therapy are hemophilia, cystic fibrosis, Parkinson's diseases, and many more. Diagram shows the steps involved in the treatment of ADA deficiency. This is the bacterium carrying the plasmid, which is cloned with the normal human ADA gene, and the genetically disabled retrovirus. Cloned ADA gene is incorporated in the retrovirus. Here, the T cells with disabled ADA genes, which are isolated from the deficient child or embryo, are extracted. Retrovirus is allowed to infect T cells, so that the ADA gene from the retrovirus got transferred to then those cells are grown in the culture medium to ensure ADA gene is active. After ensuring this, the cells are reimplanted back in the child or in the embryo. The next application of biotechnology in medicine is molecular diagnosis, which helps to solve the problem of early diagnosis and treatment of disease. Using conventional methods of diagnosis like testing serum and urines, early detection of disease is not possible. To overcome this problem, some molecular diagnostic techniques were developed to provide early detection of diseases. The first one is polymerase chain reaction, which helps in the early detection of disease or pathogen by amplification of the nucleic acid, as low concentration of pathogen or their nucleic acid does not allow its detection. So PCR amplify nucleic acid of such pathogens even when their concentration is so low. PCR techniques can also be used for detecting HIV in suspected AIDS patients, genetic mutation in suspected cancer patients, and in identifying genetic disorders. This diagram shows the PCR technique DNA which need to be replicated. The blue thing here is the nucleotide and the red is DNA primer. First, we need to separate the two strands of DNA, which is generally done at high temperature, and the process is known as denaturation. Then the DNA primer attaches at the 5' position. Next step is maturation of DNA. Here the primer adds nucleotide 5' to 3' direction, and here from 5' to 3' direction. As you can see here, two DNAs are formed, then again they are replicated, then again DNA primer attaches at the 5' position, again they mature, they start adding the nucleotides, and likewise the process goes on to form the multiple copies of same DNA in the culture. The next process of molecular diagnosis is enzyme-linked immunosorbent assay, ELISA test. It is based on the principle of antigen-antibody reaction. The infection by pathogen can be detected either by the presence of antigens or by detecting the antibodies so formed against the pathogens. It is first used in detecting autoimmune related antibodies in patients with autoimmune diseases with the help of linked enzymes. The reactions between antigen and antibody could be shown in a specific color. That is why it is named enzyme linked immunosorbent assay, because ELISA test is related to immune and enzyme. This diagram shows the procedure involved in the ELISA test. Here, the antigen is absorbed onto the well and ELISA plate in coating buffer. Then the buffer is removed and plate is washed. After that, buffer containing unrelated protein is used to block the free sides of the well. Buffer is then again removed and the plate is washed. Enzyme, which is conjugated with the antibody, binds with the antigen. The substrate is catalyzed by the enzyme to generate colored readouts, which will be later detected in the last phase of the process. I have tried to simplify the topic. Please like the video, do share it with your fellow mates, and subscribe my channel for more videos. Thank you.